Hello, my name is Carlos Valenzuela, and I'm a PhD student at the University of Texas El Paso, UTEP. And today in this video presentation, I will be talking about fighting viruses with Schlafen11. So West Nile virus in the US. I'm going to start with West Nile virus because this is the virus that I mostly work with on the lab, on my project. So I want to tell you the significance of West Nile. So here you have a map of the continental United States. I prepared this little GIF using different maps of the CDC. And the peaks from 2009 to 2019, human cases of West Nile as well as a mosquito when West Nile has been detected. And you can see there has been an increase of West Nile cases throughout the years, and this is only expected to rise with the increase in the global temperatures and also the expansion of the population. So according to the CDC, West Nile virus is the leading cause of mosquito-borne disease in the continental United States. And there is no vaccine approved for humans, and there is no treatment for people that contract West Nile virus. And about 80% of West Nile infections are asymptomatic, which means that 1 in 5 people develop symptoms. Most people develop mild symptoms such as fever. Only less than 1% actually develop something as grave as encephalitis, and which could lead to death. So West Nile virus in El Paso, Texas. Here I have a report first in all Texas. They said that over the last 5 years there have been 1,305 cases with 57 deaths. In El Paso, where West Nile is endemic, there has been 13 confirmed cases in this report written in 2019. So flaviviruses encompass more than 50 arthropod-borne viruses or arboviruses. Some of the most well-known are West Nile, yellow fever, dengue, Japanese encephalitis, Zika, and tick-borne encephalitis. So arthropods are, I put the phylum arthropods right here so you get an idea. So mostly I will be just talk about insects because these viruses are transmitted by insects, especially mosquitoes. And in the case of tick-borne encephalitis, they are transmitted by ticks. So quick, because I will be talking about the relevance of all this, I will tie it all together. I want to talk about codons and codon bias. And so this right here is an RNA codon table. So a codon, what is a codon first? It's a sequence of 3 nucleotides. So you have UCU, and this is an RNA codon table. And you have four nucleotides. So you have, in the case of RNA, you have U, C, A, and G. And there is 64 ways to arrange this into 3-letter codes, into 3-letter sequences, or make. So there is 64 ways to make 64 ways of arranging this. I'm gonna delete this really quick. Oh, I can't. Okay, well, you just scratch it up. And out of the 64 ways, there is 3 stop codons. So there's 65 ways in which you can make an amino acid, and you need 20 amino acids to make a protein. That means there is redundancy on this genetic code. There is more. You have more than one way to make a single amino acid, and this is depicted here in serine, for example. You have 4 ways to code for serine, and if you look more broadly, you see have 4 ways to make proline, 4 ways to make threonine and alanine, and so forth. However, the cell appears to discriminate against some of these codons, and some are more preferred than others. So it's called, they have a codon bias, right? So some of the codon biases they notice is that for some reason it prefers those that end up in C and G. So they call it, in some papers they go GC3 or something, or CG3, means that they prefer C and G in the 3rd position of the codon. I won't do more detail what the result of that will be, but the point is that There are some that are used more than others, more frequently, and some that are less preferred. And those that are less preferred, I'll call to be rare codons. So rare codons have been suggested, hypothesized to have different roles, or been observed to have different roles. And so the first thing is that in order to make so you have a messenger RNA. So you have a messenger RNA right here, and then you have a ribosome. So in order for a particular codon to be detected, you need a tRNA that has in this loop a matching sequence on anticodon that matches this codon and then carries an amino acid right here on gray. So you need that first. So it has been observed that the most abundant codons, they also have a high abundance of tRNAase. They correlate. Rarity of the codons and the abundance of the tRNA or the transfer RNAs. So it is believed that when the ribosome is going through the messenger RNA, which I will depict over here. So imagine that this road is the messenger RNA, and this carries the ribosome. See if I can make this work. So if they go, because they go at the same speed at even though it's like all curvy, the car goes at the same speed. But if you introduce speed bumps, and this will serve as an analogy for rare codons, the car has to slow down every time that it goes through a speed bump, as you can see in the auxiliary animation. So now that it crosses the same similar ribosomes, when they context stop codons, they make little stops, so they go slower on those sections. So there is two things that can happen. So first, the codon variants might affect protein abundance, meaning that because the ribosome is going faster on the messenger RNA, and when you have rare codons, it will go slower. You will actually make more protein where you have an optimized transcript or a transcript that use more common codons versus one that has the use utilizes rarer codons. So here use any protein really, and just to visualize this thing that's happening. So codon bias might control protein abundance. Since elongation is going at the same time as you go into the messenger RNA, so let's have messenger RNA and you have a ribosome right here bind to it. You have the polypeptide growing like this. So when you encounter rare codons, it slows down and gives chance for the protein to fold. So you have the protein and then it's folding actually. It's making a fold, and this is to work properly. If you go too fast, you might lose the protein function or structure. So here in this cell that is infected, we have 20 out of 30 particles are infectious. So we say like this, in this example, these particles have a high fitness. So these viruses have a high fitness. That means that this particle, I put like non-infectious particles on red, non-infectious, and those infectious particles on green, infectious. So the virus the population has a fitness. So fitness just refers to the ability of a virus to actually successfully infect a cell. So the higher the fitness, the more likely is it to infect a cell. Lower fitness might not infect it. So this codon variance allows proper folding, which makes correct structure, which means a higher fitness. And notice a study that I put hepatitis virus. It's called fine-tuning translation kinetic selection driving force codon variance using hepatitis A virus. And what they did on this paper, I won't show you any figures, but like they pretty much did, they saw this hepatitis A virus have a very poor codon adaptation. That means it prefers rare codons, and they thought like if we improve the codon adaptation to something more akin to what we see on the cells, then the virus will reproduce better. So they did it, but actually the virus reproduced worse. It lost fitness. It became less infectious. So it appears that this codon bias helps protein folding, also helps with fitness of the virus. So here we have that 4 out of 30 particles are infectious. So it's a very, very reduction of infectious particle when you change this. And later on they saw that if you continue passaging this virus, it will de-optimize to recover the fitness. So it's very interesting the role that codon bias has on pathogenesis of certain viruses. So control tRNAase and control translational kinetics. So, and from the case of Schlafen11, which is the protein I'm working with, there was this paper that described that Schlafen is able to specifically cleave class 2 tRNAase versus those that are class 1. And the difference here is very simple. You have a long variable loop. So it seems that Schlafen is able to somehow like to target these ones specifically. So first, a little background of Schlafen. So Schlafen are interferon-stimulated genes, and was first described in 1998 as possession the ability to suppress cell growth. So because of this ability to suppress cell growth, since it was German, it was the people who discovered this protein, they named it Schlafen because it means to sleep. The family can be into 3 subgroups. So we have the 3 subgroups here. The diagram doesn't depict it very well, but subgroup 1 is the shortest one, and subgroup 2 has an intermediate size, and subgroup 3 is the largest subgroup. So Slfn11 belongs to the group 3 protein, is the longest one. In human, there is a total of 4 group 3 Schlafen proteins, and there is only one subgroup 2, and this one, which is not characterized yet, is Schlafen12 like. In mouse, there is 5 group 3. And these ones are the ones that I'm interested in, this subgroup on mice. So this is the first instance that Schlafen was described to have viral protein activity or antiviral protein activity. So codon uses base inhibition and HIV protein synthesis by human Slfn11, and this was published in 2012. So first, before I go over the data, I would like to introduce you to some common techniques that we use in the lab, and those techniques will help you better understand the rest of my presentation. So the Western blot. So the Western blot, what we do here is that we load a lysate of cells. That means we get the cell opening with all the proteins inside, and then we load it in this gel. This is an SDS gel. And what the gel does is allow us to separate the proteins depending on their size. So big proteins travel less than the smaller ones, and so we can see like a difference in protein size right here. Then this is the separation step. The next step is the transfer. So in the transfer, we need to move the proteins from the gel to a PVDF membrane. This is achieved using an electric field, same as the separation with proteins. We use electric field to move the gel proteins to the membrane. And the membrane, what allows us to do now is to able to stain these proteins using antibodies. So antibodies so the reason we use antibodies is because we have a bunch of proteins that might have the similar size. So which means that it's not enough to separate only by weight, but actually we have to detect the specific proteins. So we use antibodies. Antibodies are able to detect specific proteins that we're interested in, a specific sequence of peptides. And so the antibody binds to the protein, and then we use a secondary antibody to detect the primary antibody. The secondary antibody is conjugated with an enzyme. The one I use in the lab is called horseradish peroxidase. and we supplement that densa with a substrate. In the case of HORISE is hydrogen peroxide. And the HORISE is able to do is break down hydrogen peroxide and create a luminescence signal. And this is known as chemoluminescence. So with this, now we have a signal we can visualize. So this visualization is the last step of the Western blot. And when able to see bands, in this case here, we see the bands that means the protein of what we're looking for is present. And the relative amount, if we can measure the pixels here, we can calculate the abundance of the protein. So it's very useful to know if you have if the protein you're interested in is there or not, or the abundance of that protein. The next technique is gene knockdown or gene silencing. And in this figure, there is two ways to make a gene knockdown or silencing. The first one here is using a plasmid with the tRNA sequence, and I will go over what that is. And this one is called transfection. And the second method they depict here is using a lentiviral particle. So a lentiviral particle, you have a RNA a genome right here, and it works pretty much the same as how HIV or other retroviruses work, that the viral particle goes into the cells, use an enzyme called reverse transcriptase to make a complementary strand of DNA, and this one, through the action of integrase, is able to integrate with the host genome, making this change permanent. And this method is called transduction. In both cases, they are able to make an shRNA. It's called small hairpin RNA. Is because it's able to make a loop. The messenger RNA is able to make this hairpin loop, and this is called small hairpin RNA. This small hairpin RNA leaves the nucleus and is able to bind with this enzyme called the dicer. And what the dicer does is cleaves the sequence, making this small interfering RNA, which is able to assemble with this complex RISC. So RISC stands for RNA-induced silencing complex. So what it does is once it's activated, and you have this particular sequence of nucleotides, is able to bind this to a messenger RNA on the cytoplasm and cleaves it, and the messenger RNA is degraded. So what happens is that the messenger RNA is not able to make the protein that it was supposed to make, so you see a downregulation on the protein that you're targeting with this specific sequence. So it's a very powerful tool. So in the case you have a protein you're not sure what roles in the cell, you can make a gene knockdown and take it off and see what changes. This is a gene knockdown silencing. The next one is a plaque assay. So the plaque assay is used for you to quantify how many actually infectious particles you have in a sample. So it's because using other methods such as PCR, that you calculate the amount of genomes that there are of your virus on the supernatant, for example, won't give you how many infectious particles there are because of the fitness thing. So we use a plaque assay in order to quantify the infectivity or how many infectious particles we have on the sample. So we have virus stock right here, and then they do serial dilutions, and then the step you have to do is days previous, you have to prepare a monolayer. So this is just a monolayer of cells that are susceptible to this virus, and then you put the microliters of each of the dilution stocks, for example, and then you cover this with another layer of agarose. And agarose does is just restrict the movement of the viral particles, so they just form like little circles. So the circles are actually an absence of cells. So where the one viral particle fell, it disseminated just around, like it was restricted, and just around and killed the cells around. So you see a nice spot. And in order to see this, you need to use a color stain to really visualize this. And now you can just count the plaques. And here they counted 16 plaques, and then you do some math, you do your dilution, and you know how much virus you added, the microliters, and the dilution, and number of plaques, and you're able to see, you're able to quantify how much virus stock you have. So back to the paper. Codon uses base inhibition of HIV protein synthesis by human Schlafen11. So here we have relative HIV virus titer and 293 T cells. So these cells, do not express Schlafen11. They naturally do not express it, and when you express Schlafen11 these cells, you introduce Schlafen11, you see a great diminution on the amount of HIV viral particles being produced by these cells, which is a strong indication that this Schlafen11 has antiviral activity against HIV. In the second experiment, they used 293 cells without the T. These cells are related, but they're different. And the main point is that these cells do express Schlafen11 naturally. So when you use the technique of gene silencing, of gene knockdown against Schlafen11, you see a great increase on the relative HIV virus titers, which is very significant. So then they compare to the 93 T cells, and then you see something similar, meaning that Schlafen11 has a strong role for preventing virus replication of HIV. So now they did a Western blot, and this is where codon bias comes into place. So this Western blot, they stain for a viral protein. This GAG is actually a gene that codes for different proteins. One of them is P24, which makes the capsid. So this is not important. I'm just saying that they stain for a viral protein. So 293 T cells, they say they stain it, and they see this abundance of the viral protein. And then when you put a Schlafen11 to 93 T cells, you see a great diminution of the amount of viral protein present. During the 293 cells, you start with very little because these cells do express Schlafen naturally. When you do a knockdown or gene silencing of Schlafen11, you see an increase. And here's just a control, and this is a tRNA that shouldn't target anything, any transcripts. So you use it to prove that it's not your system that is causing this phenotype, but it's actually the protein itself. So now they optimize the gene. So they call it opt, with optimization of GAG, and you pretty much see that there is no difference between Schlafen presence or absence in any of these cell lines, which gives an indication that this is a codon usage-based inhibition of protein synthesis. So later, it was shown that non-human primate Schlafen11 inhibits production of both host and viral proteins, meaning that not only inhibits transcription of viral proteins that have codon biases, but also on our genome there is some codon biases for certain proteins. So they show that pretty much Schlafen11 can inhibit both. It's not specific to viruses. So back to flaviviruses. So flaviviruses are single-sense positive strand RNA viruses, as covered before. They outperform human pathogens. And here you have a virion, and the virion has an envelope, a membrane protein right here, and this right here is the lipid envelope. And then you have the capsid here, and then you have the genomic RNA. So you show you this figure just to give you an idea what we're gonna be staining for on the Western blot. In this case, we'll be staining for envelope. We're gonna see how much envelope there is. So this paper was published by my laboratory, by Federico Valdez, and my PI, Dr. Manuel Llano. So Schlafen restricts flavivirus replication. So what he did here is here's a Western blot again, and we have a 172 scramble, and the scramble just means that he put a shRNA control like before, like I showed you in the other paper. That means that this shRNA shouldn't target any transcripts on the cytoplasm. And then he knocked down Schlafen11 in these A1C2 cell lines, and these are astrocytes. And the relevance of why using astrocytes is because they are able to be infected by West Nile in vivo. You know, like in people, actually, there is evidence that West Nile is able to infect astrocytes. That way it was used. So you see there is no, pretty much you don't see almost no Schlafen11. Maybe if you look really hard, maybe you see a little bit. Then he introduced a transcript again of Schlafen11 and he overexpressed it. So you have actually overexpression of Schlafen11. And he used this is what we call a loading control using alpha tubulin. And what it tells us is that he actually loaded equal amounts of proteins, because you could manipulate the results by not loading proteins in some wells and claiming that you loaded whatever. And this is a way for us to prove that in fact that the difference that we see here is not because loading differences, but because there are actual differences. Then he did a Western blot to evaluate the amount of envelope protein on the West Nile. So he got used virion from the supernatant, and that's why there's no tubulin here. He's used virion. And then you have scramble, knockdown, back complements. And it stands right up is that knockdowns, you have a high abundance of this envelope protein. And if you compare to tubulin, it seems like he loaded mostly the same in all of them. Scrambles, you see a diminution. The presence of Schlafen seems to affect E protein synthesis. So next he did was a plaque assay. So the first thing, he collected different time points of infection: 8 hours, 16, 24, 32, 40, 48, 72, 96. And this is used to track the viral growth. So and they use this scale. Mean viral titer is in a log 10 scale. That means from 1 to 2 is 10, from 1 to 3 is 100, from 1 to 4 is 1000, from 1 to 5 is 10,000. You get the idea. So what he observed here is that in the presence of a Schlafen, you have around 10 to the 6 or 10 to the 6 infectious viral particles. And when you take a Schlafen out and knockdown, you see a big increase, which is like 10 to the 8. So it means that there is around 100 times more viral particles being produced by cells that is like Schlafen. And that's why the title of the paper is called Slfn11 restricted virus replication. You see that thanks to Schlafen antiviral activity, you're able to reduce the number of viral particles produced per cell. So why is this relevant? What am I trying to answer here? So we know that in cells in vitro, which when we do experiments only on cells, we see this phenotype of low viral titers. We see less viral protein synthesis. However, this hasn't been answered yet to see if there's relevance in actual an organism. So in order to make experiments on an organism, we have first to identify the organism if it has something analogous to Schlafen11, so we can study it. So my project is the identification of murine orthologs for mice animal models. So I hope to find which of the 5 Schlafen group 3 in mouse is identical, has exactly the same function as Schlafen11 in humans. So I can move to animal models and actually study what is the relevance of Schlafen11 in an organism. Thank you.